John Thorson directs the Center for Pharmaceutical Research and Innovation at the University of Kentucky. Samples from the KGS stratigraphic test well in Carter County were sent to his center so he and his colleagues can search the samples for unique molecules, molecules that might become sources for useful drugs in the future. Tell me what it is that you are doing that we're specifically interested in for the project in Carter County. What is your research all about?
Yeah, right. So our research is in the area of natural products. We're interested in how nature makes unique molecules and also the activities that these molecules provide. So there are a number of natural products that have been developed into drugs. They have a number of different unique activities, mechanisms of action. And so we are looking for unusual environments to identify new natural products. And so this is a great opportunity for us because there aren't really many, if any, examples of people looking for these types of molecules in the environments that this drilling operation provided.
How did you find out about the project that the Geological Survey is doing, and why are you interested in what we're doing there in Carter County?
So the connection really came through some of the folks at the Center for Advanced Energy Research, and they helped me connect with the KGS, and because so we've been working with, particularly with Jim Hauer at the Center for Advanced Energy Research, to access some of these unique mining operations and collect soil samples there. And so they're aware of kind of what we're looking for in terms of unusual environments to basically get soil. And so that's how the connection came about.
This well is in the vicinity of 4,800 feet deep.
Right.
Why did that catch your attention? Was there any particular or specific reason?
Mostly because it's something that again, others haven't really tapped into as a potential environment for this, and so we were very interested just to basically see what's there, right? And the opportunity to sample, so in this case I think we were sampling every 100 feet or so, and then processing those samples from our perspective, which is to look at the microbes that might be there. Obviously it's very different from the KGS approach and what you're interested in, right? So I think I think it's a completely new area of exploration, and what we're finding, right, in this already. So it takes time to grow these samples and to process to identify whether or not there are compounds there. But we're finding that you see different types of growth depending upon the layers, which that to us is very exciting because it suggests that there are potentially different populations as you go down. Whether or not they're producing unique molecules, we'll still have to work on. But the fact that you see differences in growth from 1,000 feet versus 4,000 feet, that potentially suggests there could be some really interesting discoveries there.
You described the process. How long, if there's such a thing as typically, how long does it take between receiving a sample and arriving at some conclusion about the sample?
Yeah, so the microorganisms themselves, it will take, from a crude soil sample to the point where you actually have some things growing. It may take a month or two to actually, a lot of these are very slow growing organisms. And then from that point you have to basically start to transfer the individual bacteria onto plates until you get them purified, so you're looking at another month or two. Then you scale up production, and then so I would say you're looking at a window of maybe 3 to 6 months before you have a good idea of the chemical signature of some of the bacteria that may have been found within your sampling region. And so our operation in terms of natural product discovery here has been up and running now for just about a year, and the output I think has been quite impressive. Every strain that we have identified as potentially identifying novel molecules actually has led to the production of novel molecules. Not to say that they're all biologically active, but they are completely unique chemical structures, and so we're putting these in a repository, and trying to provide access to investigators here at the University of Kentucky to potentially use those in their assays and hopefully, find some interesting new activities for these kinds of molecules.
Which is not necessarily pharmaceutical, it's...
Right, doesn't have to be.
Right.
Absolutely. I mean, that's sort of our major focus, but
Mm-hmm.
it's not the only focus, right? There are a lot of these types of molecules have been developed, for example, in agricultural applications and veterinary applications, and there are other uses as well.
What specifically is it not specific? You're looking for anything you can find in the Carter County well?
Yeah, there's not. I don't know that there's anything specific, up front that you can look at a sample and say, oh, this could be really productive, right? So, our approach is you take the soil samples that are collected, we will then plate them out on various growth media to try to propagate the bacteria that are in that soil sample and purify those different bacteria, and then we essentially profile the metabolites within all of those different bacteria and specifically go after those that that appear to be making novel molecules. So then we'll scale the fermentation, produce the molecules, isolate them, characterize them, and then we start to work with collaborators on potential biological activity and that sort of thing.
What kind of places do you go looking for these? Are all of them deep? Are some of them on the surface? What kind of places?
No, so we're interested in particularly for soil, looking for soil microorganisms that are capable of producing unusual molecules, and these are found all over the world. The sort of the trend in the field is to particularly survey unusual environments. So right now, one of the bigger areas of focus is in marine environments. It's the seafloor or thermal vents and things like that in marine environments. My move to the University of Kentucky and to the Commonwealth actually, I think, presented some unique opportunities to explore some of the unusual environments here associated with the mining industry, associated with, for example, like this drilling operation. Also in Appalachia, there's significant biodiversity, and so I believe that there's just unique environments there as well, just in the terrestrial areas. But the impact of the mining operations, their underground coal mine fires, we're looking at thermal vents associated with those, actually collecting also within some of the underground coal mines and surface mines. So we're exploring in areas where people haven't traditionally looked for these kinds of microorganisms.
The average person might typically think, I'm talking a non-scientist,
Mm-hmm.
might typically think, we know as much as we're going to ever find out about the Earth because we have investigated, we've dug, we've drilled, and otherwise. You're telling me that we're finding literally new chemicals
Absolutely.
all the time.
Absolutely, yeah. Now, a lot of these are analogs of sort of structural classes that have been identified previously. So the percentage of completely novel, I guess what you would call a novel scaffold, where the chemical has been folded in a unique way and connected in unique ways, that percentage is lower in terms of identifying something like that. But even analogs of known, like we go back to, for example, erythromycin, right? We're now on the fourth generation of macrolide antibiotic development, and they're very subtle changes to the structure lead to dramatic, improvements in the activity of these molecules. So very simple analogs of known scaffolds can sometimes be, remarkably important in terms of pharmaceutical development.
So what you seem to be telling me here is that we might, maybe by luck and long-term research, have come across these differing analogs in the laboratory. But what you're saying is that we might also find them already created by nature on the surface, underground, and that's why you're looking.
That's right, and I think if you go back to like you said, developing them in the laboratory. So if you start with a natural product and you then want to make analogs, typically the kinds of compounds that we're talking about are very complex in terms of their structure. And so to do additional chemistry on them, to take them into the lab and to modify them from, you know, using a sort of a lab, a synthetic chemistry approach is a pretty monumental undertaking because of the complexity of the molecules. And so if you can find a bacteria that produces this by simply growing it, you know, you can scale that fermentation up, and you know, you basically have a renewable source for that molecule. And you know, part of the inspiration of why we do this is we're also interested in how these molecules then are made. So there are enzymes and very complex pathways that are involved in actually synthesizing the molecule. They basically are, you know, it's a manufacturing plant within the bacteria, right? We're interested in trying to understand how do they actually do their chemistry. So this is, for us, it's actually oftentimes just a starting point to then get into understanding how the enzymes do these unique transformations, how they make the molecules. We then try to engineer the enzymes to do new chemistries, and you know, so that's sort of just the starting point to find the molecule, but then to get in there and actually try to re-engineer the organism or the pathway is something else that's really been more of our focus long term.
Can you cite examples in your own research or elsewhere in which something has been discovered which ended up becoming a pharmaceutical product?
Oh, so there are many examples of microbial natural products as drugs. Compounds, you know, if you go back, examples would include things like erythromycin, and that's produced by a soil bacteria. Molecules like doxorubicin, the anti-cancer agent doxorubicin. So these are commonly used drugs that are produced originally by these microbes as either sort of chemical warfare agents, where they essentially try to protect their own environment from other bacteria, or in some cases as molecules for communication, or, you know, rendering other influences within their own environment. What we then do is try to isolate these molecules, characterize them, look at their mechanism of action, and hopefully, you know, if they're a unique mechanism of action that have an impact on disease, obviously try to use that as a potential lead structure.
Okay. Correctly, what you said a few minutes ago, once you have found something new, at least that you didn't know about before, you simply put it out there and making it available to other researchers. Right. So the results of some of this work may have nothing to do with pharmaceuticals. It might, I think you said agriculture. I mean, so these samples become available for all kinds of potential new products.
Right, and right now we're trying to keep that access local here at the University of Kentucky, right? But we are, and we haven't quite formalized the process for how to provide access, essentially, right? There obviously potentially can be intellectual property issues and all sorts of different things here, but the goal is to provide this unique repository of compounds not found anywhere else, and then you know, put it out there for people who have interesting biology or other potentially interesting applications for which it would make sense to screen these molecules for the various activities.
Okay. Thank you.
Mm-hmm.